All right, welcome to the first video of Unit 4. I'm going to be covering cell communication in the cell cycle. So this first video is going to be mainly talking about cell communication. The mitosis cell cycle stuff will come at the end of the unit. And the first thing we're going to talk about is the 3 stages of cell signaling. So how do cells talk to each other? And we divide it into 3 stages, being reception, transduction, and then response. We should hopefully be able to get through all 3 stages in this first video, but you can see the quick summary of what they are in the diagram there on the right, but we're going to jump right in and talk about signal reception. So signal reception all has to do with receptors. Every cell will have on its surface and displayed through its membrane a whole slew of different types of receptors. And the idea is that when we're trying to send messages between cells, we send chemical messages that we call ligands, and they will actually bind to their specific receptors on the target cell. So this, when we say, when we say the binding is very specific, we mean that the shape is super important. If it doesn't perfectly match the shape, then it cannot bind and it will not produce the response. And additionally, not every cell will have all the receptors necessary to respond to every signal. They'll have specialized receptors for their purpose, so not every cell will have a receptor for every ligand. They will be missing quite a few. So what happens in stage 1? Again, it is a signal molecule. Your ligand binds to a receptor protein and causes it to change shape. And it's the shape change that's going to actually initiate our next step inside the cell membrane. Again, this binding is highly, highly specific. It's not just anything can match. You have to have a very specific shape and chemistry to be able to bind to the receptor. And there are 3 main types of membrane receptors that we are going to talk about, starting with the G proteins, then RTKs, and finally ion channel receptors. So let's jump into GPCRs. This is the largest and most important family to know. If they give you a whole reaction mechanism, it's probably going to be a GPCR pathway. Not always, but here is the main idea. GTP, remember, is essentially the same thing as ATP. Instead of an adenine triphosphate, you have a guanine triphosphate, but it still has the same purpose of just giving energy, and it is what actually gives the G protein its name because it is activated by GTP. So when it has a GDP bound to it, it's inactive. If it has a GTP bound to it, it's active. And so when a signaling molecule, when your ligand binds to your receptor on the outside of the cell, it causes a shape change that will then bind to the G protein inside the cell and cause it to get phosphorylated, gets a GTP added. So it gets the GTP, this gives it some energy, and it gets released from the receptor. The receptor pops off its ligand, and the G protein with its activated GTP heads and slides down the membrane to activate another enzyme that will start to trigger the response. So it's kind of just like an on-off switch, with the G protein actually being the switch. When the switch is slid to the left and it's inactive, it's off. When it's slid all the way to the right and your G protein and your target enzyme are bound, then your switch is on and you get a cellular response. So a large portion of this is just how do we get the signal from the outside of the cell to the inside, and that is what this G protein does. It binds to the receptor and accepts the GTP when it's active. So we've then transferred the signal from outside to inside. The next main type is RTKs, or receptor tyrosine kinases. The word kinase is going to come up a good bit in this unit. When you see it, know that it just means a protein, a type of protein that is going to attach phosphates to other molecules. This tends to activate them, give them energy, make things happen. So Same idea though, ligand binds to the receptor. The only difference is the receptor in this case is actually in two parts, and you need two ligands to both bind. It's kind of like turning the two keys in the submarine to launch the nukes. You need both ligands to bind to both halves of the receptor, and then the receptor actually comes together and forms what we call a dimer, which just means a complex with two parts. And then the magic happens. So it's bound to the receptor, it's activated, and now what's going to happen is those tyrosine regions that give it its name are going to get phosphorylated. They're going to get an ATP added onto them and become active. And after this happens, they are able to then pass that phosphate group, that energy, off to further proteins that will then carry out the response. So in this case, if you remember the G protein and the G protein itself was moving to activate the next enzymes. In this case, the activator protein is staying put, and it's the other enzymes that actually come in, bind to it, get the phosphate, are activated, and move away. The big thing with this is that it can trigger multiple pathways at once. One receptor has 6 tyrosine binding sites, and so could theoretically trigger 6 different enzyme pathways to go. And because of this, if you mess with the pathway, you often get a cancer. Will often develop because of it, which we'll talk more about later in this unit. The last type of receptor is just a ligand-gated ion channel. This is just your standard ion channel that only opens when it's bound to the ligand receptor. So the ligand binds, the channel opens, and the ions, it is a specific ion, so it won't let just any ion through. But they'll just diffuse into the cell down the concentration gradient. Doesn't take energy or anything like that. So this one's pretty simple. It's actually the ions themselves that will go on to cause the response, so there's not a phosphorylation step here or anything like that. It's the ions that cause the response. And then lastly, which I didn't have these mentioned in the main one, you won't see these come up very often, but they are there. We do have some receptor proteins actually just inside the cell. They can be in the cytoplasm or the nucleus. And they are activated by ligands like testosterone, which is a small nonpolar steroid hormone that can cross the membrane on its own. So these ones, they're a little different because they're not membrane bound like our other three. But they essentially do the same thing where your hormone binds to it. It gets a shape change that causes your actual response to trigger. So that is our reception stage. Let's get into stage 2, which is transduction. So what does transduction mean? It's kind of a cascade. It's how we transmit and carry the cell from the membrane and amplify it and bring it to the areas in the cell that are actually going to carry out the response. So it's usually a multi-step pathway, and it looks very complicated when you see them. But the reason for this is to give us opportunities for coordination, regulation of the response. So we need to be able to shut it down, turn it on, speed it up, slow it down at various points. And so if we have a big multi-step pathway, it gives us a lot more opportunities to do that. Whereas if it's a simple one-step pathway, it's all or nothing. You're not gonna have much control over that response. Most of the time, these relay molecules are just proteins. But sometimes they are second messengers, which we'll talk about. And the main goal here is really amplifying the signal. You can have just a few signal molecules, just like a few ligand molecules binding to the receptor, and you end up producing a much larger response with many, many more molecules involved. So let's take a look at a sample of a phosphorylation cascade in a transduction pathway. So signal binds receptor, and then we activate our relay molecule. It's not telling us if this is a G protein or RTK, doesn't really matter. Kinases are here, and they're going to do their kinase thing and transfer phosphate groups. They're going to phosphorylate different proteins. Phosphorylating just means adding a phosphate Dephosphorylation is removing a phosphate. That's done by proteins called protein phosphatases. You won't see those that much compared to kinases. But again, it's a molecular switch. Phosphorylation will generally turn proteins on and make them active and functioning, and dephosphorylation will deactivate them and stop their functioning. So you can see here, you get your relay molecule. It activates the first kinase, which then activates the second kinase, which activates the third, fourth, and so on. And the key thing to understand here too is that each of these activated kinases can activate more than one other kinase. So each step can kind of multiply the number of molecules involved by like 10 or some amount. But you're multiplying the number of molecules involved at each step, which gives you your cascade, and then finally get your protein that will actually carry out the response at the end. There is a second form of this cascade that is not just a protein phosphorylation cascade. This involves secondary messengers. So ligands binding to your receptor is considered the first messenger. And so your second messengers are these small non-proteins. Importantly, they are not proteins, but they are water-soluble or some form of an ion. So they're not going to be able to pass through the membrane on their own, but they just spread throughout a cell by simple diffusion. And they'll just kind of fill up the cell. These mainly are there in GPCR and RTK pathways. And there are two main ones to know. The first would be cyclic AMP, which you'll often see as CAMP, CAMP. And then you have calcium ions as well. And these are second messengers. They will actually just do the job of activating proteins instead of a protein kinase. There might still be protein kinases involved, but in the example you can see on the right, it's a G protein pathway, and the G protein actually activates adenylyl cyclase, which makes cyclic AMP. And so the idea is just when you have the signal active, you make a bunch of cyclic AMP, and that's going to go on to carry out other responses. So this would be another way to get a bunch of different responses all at once. Stage 3 response, pretty simple. This is this time when the cell actually does something with it. Most of the time, it's turning on a gene or turning off a gene, meaning telling it to make a protein or not telling it to make a protein. But it could also be, we could also be amplifying a signal. And this is where we can also make it more specific, tweak its efficiency, and ultimately where we will end the signal. So let's talk about the signal amplification and its specificity. Again, the enzyme cascades have amplified our response. So we now have a ton of molecules involved in this response. And so the other things that can happen here are that when you're thinking of the responses, your pathway, you could have the simplest form where one molecule leads to a simple single response, no branching at all. But sometimes you can branch into multiple pathways, like you can see in the second panel up there, where one ligand will actually have multiple responses. And sometimes you have multiple ligands that both coordinate a single response. So you might have a ligand and a pathway that only serves to activate or inhibit a separate pathway. And then sometimes you can have the same ligand but a different receptor, and so you get a different response. So there's a lot of different forms of response beyond just telling it to make a protein, because it could also just be involved in tweaking or messing with the other response pathways of other signal transduction pathways. And then finally, we end the signal. This is where the signal fully stops. Our ligand concentration falls, fewer receptors are bound, and ultimately they revert to an inactive state and the response stops. So now that we've gone through the steps, let's quickly talk about a few cell signal pathways that are important. The first being apoptosis. So apoptosis is your programmed cell death or cell suicide. In this process, the cell is going to kind of chop up and package all of its components and put them in vesicles, and then it will be digested by scavenger cells around it. It'll kind of pinch itself off and give an indication that it's dead and wants to be digested. This is really important for in animal development as well as just general maintenance of our tissues. If cells become defective, we don't want them to continue dividing and existing. So there's a few ways to trigger this. It can be an extracellular signal telling it to do it. It could be triggered by DNA damage from the nucleus or protein misfolding in the ER. But once the response is triggered, it is ready to go. And interestingly enough, the default of the apoptosis pathway is to kill the cell. There's actually a protein embedded in the mitochondria membrane that permanently basically deactivates the apoptosis protein pathways. And that protein gets deactivated by a signal molecule, which then lets apoptosis proceed. So like by default, apoptosis wants to happen, and our cells are actively stopping it. If our cells can't do this, let's say that CED9 protein is super overactive, then you can get cancer because a cell is unable to undergo apoptosis. Another example where it's used is in the formation of our fingers. So essentially we grow a full mitten first, and then our cells get signals to die off in between and actually form the fingers, so that space gets formed afterwards, and is just left over by the cells killing themselves off. Another important one is with diabetes. Diabetes is involved with our insulin pathway, which involves glucose uptake into our cells and glucose storage. So the two forms of diabetes, type 1 is generally the more genetic one, and that is just you can't make insulin, or the insulin you do make is defective. And so nothing ever binds to the receptor at all. There's nothing to bind to the receptor, and so glucose ends up staying in the bloodstream. Type 2 diabetes, the less genetic one, more environmentally based, you are making insulin, but it's almost like an insulin resistance. The insulin essentially just has trouble binding to your receptor or staying bound to the receptor, or the receptors are just not really producing much of a response when they are bound. It's like you're developing a super high tolerance to your own insulin. And so again, the result is glucose stays in the bloodstream, but it's for a slightly different reason. But many, many diseases are a result of just a malfunctioning signal transduction pathway. And that is going to be the end of the first video. It is a longer one. I'm sorry. I tried to shorten it, but it just didn't really lend itself to shortening. So I'll shut up and let you go. That's all. See ya. The next one should be shorter.